go ahead and start speaking. And if anybody joins us, I can let them know that the recording will be available afterwards.
Okay, sounds good. Yeah.
All right.
Yeah, so we have at least currently 2 students that I'd like to hear a little bit more about like your interests and what kind of what you're looking out of the course. But maybe I can kind of give a brief overview. I can start with a brief overview of what the course, as I'm thinking, should be about. And then, like to hear more about kind of what you are looking out of the course. And then there will be, changes. It's still a little bit volatile in terms of the kind of final structure of the course. So actually your feedback will be quite valuable because I would be able to incorporate some of that. But maybe I can start with kind of how, what I would propose, and then I'll take questions from you guys, depending on where you also are. Does that sound reasonable?
Yeah, sounds good.
Okay, cool. So then let me also share the screen since it's always a little bit easier. All right. Can all of you see my screen? And with some writing on it. Okay, so yeah, the plan is, so I am a new faculty here in between the bioengineering department and the cellular molecular medicine. And I specialize on a very particular form of imaging, a type of multiplexed imaging related to this new wave of spatial transcriptomics. And as you'll see, it has some interesting intersections with live cell imaging. And I'll be teaching this course it is entitled Molecular Imaging and Quantitation in Living Cells. And I think my plan for this course is to also focus quite a bit on just the molecular imaging and quantitation, as well as what tools are available in living cells and kind of how you merge information from living cells to things in fixed cells, particularly in light of some of the new imaging modalities that exist and might be useful for you guys to know. Those include spatial transcriptomics, which is quite popular, and some of you might have already heard about it. So yeah, so the course is gonna have a few objectives in terms of what it aims to achieve. So one of the first things that would be useful is to kind of get everyone up to speed with some of the basics of imaging. Maybe it's even more obvious if I kind of look at roughly what topic we're going to cover almost lecture by lecture. So we're going to talk about some of the fundamentals of optics. I want to make sure that at the end you'll know what a lens formula is, what diffraction roughly is, and some of the basic properties that are kind of essential for anyone doing microscopy to kind of know. I will probably also, I'm thinking of including some information that is just practically very useful as well, as hints here, like what is an objective, kind of use an objective and so forth. Then it kind of very related to this, obviously, some of the information on microscopy and how some of the fundamental things in optics is applied to microscopy, including some of the basic microscopy modalities, going from confocal to a light sheet microscopy to some of the very basic type of imaging modalities. And that's going to sort of continue along the same line up until where you see blue. These are, by the way, very similar to the course previous years. So you can also ask previous students, or you can see previous material. This is going to be almost identical to that, well, similar to that. And this is going to cover pretty much roughly the same topics. With slight emphasis here and there. And then this will, of course, yeah, and this will include both some of
I sent you this. It's like,
yeah, great, thank you. Yeah, and this will include some of the, kind of basic fluorescence up until some of the a little bit more modern, both in terms of rough life imaging modalities, as well as spatial modalities. So kind of up to here, pretty standard. Now, where it's gonna separate from the course last year is the focus is not going to be only on live imaging. One of the very interesting directions that is currently emerging, particularly with some of these advents of kind of large and powerful neural networks, is on this interface between doing some type of imaging in live samples, fixing those samples, doing a whole collection of other measurements in the fixed samples with even more other powerful modalities that weren't previously possible before, and then integrating this data and kind of enhancing both datas. So because I think that's a super important direction that is now flourishing, I'm thinking of having some of that in that course. So in other words, I'll be talking about how to extract a lot of information from core images. So I'll be describing what spatial transcriptomics means, which is one of these famous new modalities of how to extract, to effectively do many, many, many colors in a single fixed sample. I'm going to go in a little bit more depth of one of particular ones that I specialize in. And I'm going to explain how this ties to CRISPR. So to some of these other live modalities that are kind of at the interface between the two, live imaging and more kind of programmable fixed imaging. Okay, cool. So up to here, this is kind of the major core part of the course that is mostly lecture-based. So should give you background on from basic things to the more modern things. And then afterwards, everything is gonna be a little bit more oriented towards an actual practical application. There'll be, similarly to last year, a practical component to the course, which will be an experiment. It'll be a different experiment than last year. I thought it would be fun to have an experiment in which kind of the spirit of the course, as in have a sample, very simplistic sample of a few cells that are kind of crawling on addition, dividing, image them a few time course, likely primarily their nucleus, fix the cells and do a form of simplified Burfish, simplified form of just measuring a few single molecules, single transcripts in them, and then remerge that later information with the original information of where the position and likely the lineage of the cells were. A lot of that will be even more obvious as we kind of go into it, but it'll be an experiment. It'll be kind of on this interface between, I guess, high spatial content information merged with some information captured from the live imaging. And a lot of it would be here. That's kind of basically it for the overall layout of the course. I can later talk about some of the things that you might need to do for the course just for your credit, but are there any questions at this point about the rough direction of the course? That was a copy while that was brewing. So maybe to kind of stimulate that, people that are on, maybe we can kind of get to introduce each other just so know a little bit more about, if you're considering taking this course, also what you might want to achieve out of the course. Does anyone kind of want to describe a little bit about what they're doing and why they might be interested in the course? And we can kind of go from there. No? Yes. Debalina?
Yeah, sure, I can start. So I'm a PhD student at the chemistry and biochemistry department, and I am working on what I consider as an alternative to CRISPR, where I'm working on developing tools that can edit other RNA molecules, but it doesn't use CRISPR, it uses an RNA to edit other RNA molecules. So at some point in my research, I think there was a talk of maybe using facts um, to support to get some data on my research. So um, I've been interested in like for a while in fluorescence microscopy. So the moment I saw this email about this course, I thought I'd come and check it out and see if taking this course might help me.
Okay, good. So that's a very good point. So indeed, facts of flow cytometry is kind of a super important thing for a lot of people, partly because of how easy it is to apply to cell culture and the quick information that you get back. So we're going to discuss it. And, the good thing about it depends how many students we'll have, but we're not going to have students that are going to be too likely not. We won't be overwhelmed with students, then I'll be able to kind of focus on including the needs that you might have, and we'll have discussions on that. So for fact sorting, I'll be able to provide both some information from my perspective of how that could be useful, and we could have like a discussion as well. I think that's true for almost all of these modalities. One of the things that I would want to encourage you is to actually think, is this useful for my research or not? Is this available or not relatively close by? And the answer might actually be yes to both of them, in which case, you'll get something very useful out of the course. Another thing that, for example, Dabalina could be potentially useful for you would be something like single molecule fish, which is now getting increasingly easy to get access to. So by that I mean, you have a sample and you're manipulating in some way the RNA within the cells. You might want to know exactly what's happening to that RNA and count it relatively fast with very high resolution, right? So very accurately.
That is exactly what I'm looking for.
Right. So for that, this course will teach you how to do pretty much like experimentally how to do a single molecule fish, such that you'll be able to quantify that particular RNA that you'll want to work on. So far, this was designed to kind of give a sort of more broad, just kind of show you the protocol, how to do the quantification and everything in a very particular type of sample. But, obviously we'll want to discuss that. If you want something that is very practical, we might actually want to bias it towards that. So I'm open on this experimental part for being a bit creative for the needs, if there are some clear needs, especially since many of you, all of you are going to be graduate students. So I know how that's going to be important.
Okay, thank you.
Cool. One thing that I should mention is for some of this, including for fact sorting, there's going to be there's always a computational component when it comes to imaging. One thing that I actually I stress quite a bit because I think it's immensely important is to be able to extract some sort of quantitative information from the images that you'll acquire. So for that, the more modern way will be to use a kind of widespread programming language like Python. So this course is going to introduce you to some of those basic sort of Python libraries and imaging modalities that you could use. That might seem a little bit daunting, but you shouldn't worry because to some degree, it won't really be part of your grade, so you shouldn't be worried about that. And however much you'll get is going to be just good for you. I can say that from my experience, 2 years back, I was a graduate student, and one of the things that I appreciated from a course that I took, a machine learning course that I took, was actually some of this core just infrastructure to know, like how to open Python Jupyter notebooks in a particular context, and things that you'll learn, for example, is what is Napari, which is the equivalent of ImageJ for Python, and it's hardware accelerated, so you can open an image in a more modern way, actually be able to process it. So kind of things like those are going to be underlying it, at least kind of starting from here onwards, which could be generically useful for anything to do with imaging, even things to do with like basic programming. having a little bit of an infrastructure here should be kind of very generically applicable. Okay. Any other taker? You want to introduce yourself? Maybe ask any questions?
Yeah, I can go next. So hi everyone, my name is Deepak. I'm a second year PhD student in biosci. I'm from Genial's lab. So like our lab does a lot of things that are related to like spatial methods, spatial transcriptomics. So this course does seem like it might be a good primer for me to get more involved in those things in the lab. So, and I think what I'd be interested in is the programmable live cell imaging using CRISPR. That part seems really interesting to me. I mean, everything is interesting because like things like storm imaging, palm imaging, just to know on deeper level about how they work. Those are all interesting things. And yeah, live cell imaging is also something that I'd be interested in. So maybe
Yeah,
like more practical applications of like, I mean, it's important to know the basics about that thing, but also how you practically do it. I mean, it's not that easy to probably learn in like a day, like an hour, 50 minute, but yeah, those are the things that I might be interested in.
Yeah, I mean, I think so, you know, I think even from a historical perspective would be interesting to know. So let me kind of, the reason it has the structure, it's not, it actually has a very good purpose, which is some of these spatial transcriptomics, particularly this one that is called MERFISH, which is kind of a conjugate to some of the ones that are being developed in Jinyo's lab that are a bit more sequencing based, will give you a little bit of interesting other perspective. So this particular method that is called MERFISH, which is one of the prevalent types of spatial transcriptomics, was developed in the same lab as the STORM method, the super resolution method, and it's my parent lab. And in which is Xiao Zhang's lab. And the reason it was developed by that is because there's actually an intimate relationship between, you know, how do you do super resolution imaging? How can you take some of those concepts and bring them to multiplex imaging, which is MERFISH? And how can you take those components and push them even more to live cell imaging? So this is kind of the very frontier of where people are kind of trying to go, at least on the technology side. Very difficult, but you know, it kind of give you some, hopefully some sort of perspective on how that is also being developed with some background of all of these techniques, which hopefully will be useful.
Yeah, for sure. Like I learned a bit about Storm and MERFISH when I did my rotation in Pallav's lab. He's also, I think, from Zhao Wei's lab. So that's where I got to learn a bit about MERFISH.
So yeah, so hopefully this will give you, yeah, exactly. So I'll keep that in mind, and I'll see how we're organized and boost this, particularly as we'll have more conversations.
Yeah, what would be nice is like for these things that are sort of more at the front, like just be knowing just a primer of what is like currently happening, like where have people, what advances have been made in terms of maybe live cell imaging or any of those imaging methods. That'd be sweet.
Yeah, I agree. I will keep that very much in mind, right? Part of you know, you guys are scholars, so I agree. You should definitely kind of know where that's why I kind of wanted to include these, because there is one of the frontiers of live cell imaging, and they do sort of come from roughly this direction. So it'd be nice to know this direction and know at least this particular frontier that I think would be interesting. So I completely agree.
Thank you.
So we'll discuss some of the papers. That actually brings an important point. Obviously, some of these are textbook materials, so they're just kind of fundamental. The moment they advance in the course, like Storm, we'll discuss, it will be based on the paper that had Storm, and the same for MERFISH, the same for these programmable CRISPR. So it'll be very quick onto like where the state of the field is and what are some of the important publications and what's the most cutting edge. So I think that makes sense of the course.
I also have like one suggestion. Like when I read about all these methods, I feel like it's hard to then sometimes draw back on what fundamental methods these, like for example in MERFISH, like is it confocal microscopy? What sort of microscopy is like underlying those methods? You know what I mean? Like sometimes when you look at all these methods at a very big scale, you forget, it's hard to extract what's the basic underlying optics that's happening between. So like whenever you explain these methods, it would be nice. Yeah, what's the basic underlying? What's the basic underlying technology and like the optical microscope type that you're using for those things that people use for those things?
Yeah, I'll do that for pretty much all of them. I mean, the nice thing about we'll be able to reference based on what we'll discuss before. That's why, for example, like for merfish, do you want to use a confocal microscope or not? Or for live cell imaging, what is the best microscopy? Should you be using a light sheet or not? And what are the trade-offs? And kind of what do people do in the most common interface? I'll keep that in mind. I think that's a very good point. Okay. Any other questions? Oh, I did wanted to say one more thing before that, which is in terms of credit, I think the way I want to make it as less stressful for you guys as possible. Then I thought a good way would be to just have roughly how it was last year, in which attendance is obviously very encouraged, but won't contribute that much to being penalized, since I know you're going to be busy. However, things that are going to matter are a presentation and kind of a report. By report, I really mean more like a oral presentation. There are going to be two types of presentation. It's for a project that you kind of would imagine doing. So kind of a proposal of a project and a presentation of that proposed project. So for example, since kind of Deepak mentioned a few things, or Debalina mentioned a few things, a very natural thing would be, I would really want to do, let's say, fact sorting for my cells, and I would consider doing some single molecule fish to be able to quantify my stuff. Okay, how can I write that down in a way that is, useful, such that it's very clear what the target is, given what I've learned from the course so far? Or alternatively, I want to apply some of this CRISPR technologies for this particular problem. Let's say I have some, cancerous GBM cells, and I want to, monitor something during a treatment. This is kind of how I would propose to do it. And that way we can also discuss both the feasibility of that, as well as the, kind of some of the underlyings. And then it will kind of force you a little bit to think whether it's feasible or not, and whether you kind of comprehend some of the components, or what components you don't comprehend. And then, after you'll submit this sort of proposal. It's kind of like a proposal of a grant, in which you'll kind of have roughly the same components, and then you have a presentation on it for what you'll want to do. And that's all that is going to contribute, the way I kind of think about it, because that's sort of the thing that you'll really be doing in reality. And that way it will kind of make you think of whether any of this would be useful to incorporate or not. So at least we'll make sure that at least the core fundamentals you'll have some comprehension on is the ones that are most relevant for the type of things that you want to do. So very encouraged to focus, to see if it's connected to your research. It doesn't need to be, but if it's obviously a plus, if it is, because then it might be even more useful. Okay. Anything else?
I can go next. I'm Nari Wong, a second year PhD student from Julian Schroeder Lab. So I'm studying plant. Basically, I'm interested in how the guard cell, which is like a plant mouse on the leaf surface, respond to high CO2 and high temperatures. And the reason why I'm interested in this class, because I would like to develop a sort of biosensor that, for example, like FRET-based biosensor that can probe the interactions of the CO2 or bicarbonate sensing component. So I wish I learned the stuff like the basics of the microscopies and probably the development of some FRET based sensor, something like that. Yeah,
sounds good. So I'll keep FRET as part of the core components, because it's still, at least from the continuity of the previous course, I'll remember to kind of keep that as one of the core technologies that we're gonna put. It's gonna, so yeah, it will be a little bit less so focused on FRETA as it was before, partly because the previous instructor was one of the very leading pioneers of the FRET technologies. I am a little bit more focused on kind of the spatial distribution organization within cells. So there's gonna be a little bit of a shift, but nevertheless, we'll go through all of the kind of core microscopy elements that you would need to be able to do that as well. So I'll make sure that there's a good balance between the two things. Cool. Any other questions, concerns?
Could you explain what you mean by spatial distribution of organization of cells? I'm new to this field, so I've never done like fluorescence microscopy in my life. So I was just wondering, like, are you looking at like the organelles, or are you looking at how two molecules are interacting within the cell? Like, what does that mean?
So I can explain a little bit. One of the very exciting recent directions of imaging has been this ability to image with a lot more colors than ever before. And that's kind of a very fundamental thing, right? So it's like if you have a sample, like a cell, ideally you would image that cell in more than 5 fluorescent colors that is on a normal microscope. Now, if it's a fixed cell, the way people have been imaging is you take your cell, you fix it, right? So you add formaldehyde to it and it's fixed. And then you, rather than imaging with fluorescent colors, you add some sort of, you connect it to a fluidics device and you just, sort of flow and stain different things across different rounds. And you can do up to like 1,000 colors or so of, for example, different species of RNA. And you can measure within a cell different mRNA molecules, up to 1,000 types of mRNA molecules, or even more. And then know exactly where they are in the cells, what they're interacting with. For example, they might be interacting with a particular type of protein, which you also co-label. Or what I specialize even more is label DNA pieces and see if, for example, different enhancer elements interact with certain genes and whether that promotes transcription or not. And all of these kind of the fundamental technologies to be able to take a cell and image in it all these different components, all in different colors. And then the whole question is how to do these many, many colors. And perhaps a question that is sort of somewhat relevant to this course is, can you do that in live cells, and how much can you push some of the concepts that we've very recently learned towards the live cell? That's sort of the core of it.
I see. Thank you.
But yeah, I mean, a lot of it will become even more clear as we kind of progress through the course, particularly when we get to somewhere in this region, which I think is going to be, well, I hope it's going to be at least a little bit, new to some of you, so you will know kind of where some of the leading microscopy is, at least one of the directions. So, there'll be some novelty to it. But hopefully some of those components will also be useful. As I said, some of them I can immediately see how it would be useful to including you, Depolina. Like if you are thinking of, even for quantification, like if you're thinking of, I have some RNA or I have some proteins that I want to measure relatively, accurately, or I want to measure the interaction of a protein and an RNA relatively accurately, how do I do that? And some of it might be live, some of it might not necessarily need to be live, and there are different ways to do that. So hopefully this course should give you a sense of how to achieve that.
Okay, thank you.
Okay, sounds good. Anything else?
I was wondering for the lab modules, like, is there any point where we would actually get to see the instruments to see how they're...
Yeah, of course. That's the exact whole point of them, indeed. So that's a very good point. Sorry, I didn't
It
really
says
explain it.
video presentations. I kind of got confused at like it says video presentation and discussion.
Yeah, I
Yeah,
inherited this from the previous one. I don't know what that means. All right, so actually, this is a little bit, sorry, I didn't update this properly, so my apologize for these. This I just forgot to, I'm not on the most recent version, I apologize for that. But I can explain, maybe the best is to explain just verbally. So this is what I envision, depending on number of students, but I think no matter how many number of students will be able to accommodate based on groups. I envision just booking 4 microscopes that I currently have in my lab that are capable of doing kind of some of these technologies. So some of this, a little bit of this, but mostly focus on this. And then just use those to first just image a few cells before applying a simplified version of this particular method, and then imaging them afterwards and integrating the information. But that would mean that you would come, you'd see both the microscope, explain roughly how to operate it. You'll be able to operate it for this very particular task, take some images, and then analyze some of those images. That's roughly what is really going to be. As you see, I'll try, I currently pushed it for quite a reasonable number of days. I'll see if I need to push it for a little bit more. But yes, I do want you to actually get to play with a microscope, and particularly a relatively modern microscope that you know, can do some of the things that are going to be presented in the course.
All right, thank you.
Good. Yes, sorry, I forgot to explain that clearly. Good. Okay. I mean, if there are no more questions, what do you think, Kelly?
Pardon me, wanted to make sure that it was okay with you if the students do end up having some questions, that they reach out to you and contact you on email. And yeah, there you go.
Yeah, of course, right? So that's a good point. Obviously, you know, some of you might not see this live, so feel free to contact me about everything regarding the course. I'll be obviously try to be very responsive. And yeah, and of course, right, we'll go from there depending on everyone's needs. If you have feedback and you want, you know, like you, again, if it's something similar to how you describe right now, like I am doing this research, I am curious to know if this course would be useful for me, I'll be able to write something back similarly to how we had all of these discussions. So that would be one set of questions. Other questions I can answer, just organization questions if you want to know final project, what it mean, how much time do I need to dedicate, things like that. We can also discuss that.
Excellent. And I'll be sending out an email to the whole interfaces community to say that this video is available. So some students may watch it and after that maybe have some questions. But I think this is great. I think it's going to be very helpful to have for it to be on our YouTube site for students as they go through their academic year, you know, in case they didn't have a chance to see this. So thank you so much for attending, Natty Wong and Debelina. We appreciate it. And thank you so much, Dr. Bentu. And y'all have a great rest of your day.
Perfect. Thank you so much, Kelly, for organizing this.
Thank you. Bye-bye.